There might be a few stragglers coming in, but I think I'll get started now. So I first want to say thank you to the organizers, if any of them are here, for the opportunity to present today. And I'm really honored to be able to speak in front of such a courageous and wonderful group. So it's really wonderful to be here today, and I hope that I can offer not only an introduction to Skyhawk and our lead compound for Huntington's disease, but also some general biology background that might help as you're exploring this space. And given that there are so many wonderful opportunities coming forward for therapies, potential therapies for Huntington's disease, hopefully some of what I talk about today will help cover the aspects that are being discussed across various different therapeutic modalities and approaches. So I'm here to introduce Skyhawk Therapeutics and our lead compound, SKY-0515. This is a daily pill designed to target the cause of Huntington's disease and with the goal of potentially slowing the progression of the disease. Before I move to the next slide, I just have a fun fact question for you. Does anybody know where the name SKY-0515 comes from, related to Huntington's disease? So May 15th is the International Awareness Day of Huntington's disease. So we have named our lead compound, SKY-0515, after that day. So my disclosures are just that I am a full-time employee at Skyhawk Therapeutics. And just as a very quick introduction to Skyhawk as a company, so Skyhawk was founded in 2017. It is a biotech company based out of Waltham, Massachusetts, outside of Boston, as well as Basel in Switzerland. And our general objective is to develop small molecules that modulate RNA splicing. And through modulating RNA splicing, this can control the production of proteins and proteins that are toxic to the cells. And SKY-0515 is just one of these molecules which we have developed that is a daily pill. So it is a small molecule. It is a daily pill that is taken orally by mouth once a day. And sorry, I'm going to walk to this side. It has the potential to slow the progression of Huntington's disease through targeting the RNA of Huntington, but also another RNA which produces another protein, PMS1. After targeting the RNA, the function of the compound, it works by reducing the levels of the harmful protein that are produced in the cells. So in the case of Huntington, we know that we have, in Huntington's disease, a wild-type allele and a mutant allele. And our compound will target both, so we will be reducing the protein of both wild-type and mutant Huntington, as well as, to some extent, PMS1. And I'll talk more about PMS1 in the future slides. We have finished a phase 1 study in healthy volunteers in Australia up to 14 days. And during that study, we were able to demonstrate the amount of lowering of Huntington RNA in the blood, which I'll show later. But we were also able to demonstrate that during that duration of time, SKY-0515 was well tolerated. Okay, so before I go more into SKY-0515, I wanted to just back up a little bit and talk about the different modalities or types of medicines that are being developed for Huntington's disease. This comes into some of the stuff that you heard from Rachel this morning in terms of different approaches that are being pursued. Many of them are Huntington lowering approaches, but not all of them. So first of all, the small molecule class of compounds, which includes SKY-0515, these are very small chemical compounds that are synthesized in the lab They are generally manufactured as a pill and can be taken orally. And they often can be designed to spread easily throughout the body, including into the brain, so crossing into the blood-brain barrier and reaching the brain and even deep structures of the brain. The second class of molecules, which you may be familiar with, are nucleic acids. These are molecules such as azos, which are, you might have also heard as antisense oligonucleotides, or siRNAs. These are what we call medium-sized class molecules, and they are made up of nucleotides, which are the components of your DNA and RNA. These are also synthesized in the lab, and they are small stretches of nucleotide sequences. Due to their size and properties, they cannot be taken as a pill, and they generally need to be injected either into the blood, specific tissue, or spinal cord for access to the central nervous system. They don't generally spread as easily throughout the body, and they may have trouble reaching some cells, in particular deeper regions of the brain. The third class of molecules are the biologics, and here there are various different types of biologics, but I'll just highlight two, and those are antibodies and viral particles. The viral particles are the vectors that are used to deliver the gene therapies in the different talks you've heard about today in terms of gene therapy. These are large molecules, and they are produced using living sources, in particular cell culture. And they must also be given by injection into the blood, specific tissue of target, or the spinal cord. They often do not spread as easily throughout the body and may have trouble entering cells. However, there are technologies that are being developed to ease that delivery to specific cell types, including in the brain. So now I want to talk about RNA. Because that is really where our bread and butter is and where we focus on our, in terms of our therapeutic approach. So you heard a lot from Rachel this morning, and I'm actually going to use a lot of the same terminology and the same analogies that she was using, but repetition is good, so hopefully this will not be too redundant for you. So RNA is essentially a copy of a piece of our DNA, which is our permanent genetic code that every cell has. And it is a copy of, for example, one gene that gets carried throughout the cell. It gets modified further, and we'll talk a little bit about that in a minute, and it gets produced into proteins that then are used to make the cell function and healthy. So you can really think about our DNA as a very large cookbook of tens of thousands of recipes. And then the RNA being one of those recipes which has been copied from the DNA, from the gene. So one gene is you make a photocopy of that recipe, and that photocopy is then transported over to the stove to produce a dish. And in the case of the RNA, that first copy that gets made is a full recipe, which includes not only your ingredients, but it also includes more information about the dish, maybe something about a little bit about what that type of ingredient is, how you might want to potential modifications of that ingredient. So there's lots of little intermittent steps, and I want you to remember that because that goes into the processing of the RNA, which I'm going to talk about in the next slides. But this is essentially the process of going from the DNA to the RNA to the protein, which you're probably quite familiar with at this point. So what can a drug that is targeting RNA do to help modify disease? So essentially drugs that target RNA, which are small molecules like SKY-0515, but also antisense oligonucleotides and other approaches are being explored which target RNA. And they generally interact at a specific site on the RNA to modify how that RNA functions. And that means they could change the function through either stopping the RNA from making proteins altogether or providing instructions to modify how that protein is made and potentially modify the function of that protein. They can modify the stability of the RNA, meaning they can make RNA last longer or potentially degrade faster, thereby changing how the RNA dynamics function in the cell. And ultimately, they can influence what we call gene expression without permanently altering the DNA. So I want to come back to this process, this idea of processing of the RNA, because there's one step that I left out during that, that diagram of DNA to RNA to protein. And that is the step between pre-mRNA and mature mRNA. And during that step is where splicing comes into play. And so when you make that photocopy or that copy from the DNA or from the recipe book, as I said, you have the precise components of the recipe, the ingredients, listed out that you need to make that protein. But you also have other things written in that recipe that give you directions or things that are not necessary for that dish to turn into that, for that whole dish to turn into your final product. And so these are the pieces that you want to cut out when you're actually making the dish. And this is what we call splicing. So in DNA, or in RNA, which is first transcribed from DNA, you have this pre-RNA that has a much longer sequence, which includes what we call exons and introns. And the exons are the protein coding portion of the RNA, and the introns are the part that we want to remove because these do not inform what needs to go into the protein. They do offer important aspects for the processing of the RNA, but they need to be removed before you can actually get the good protein to be formed. And so splicing is where we come in with SKY-0515. And so this is just coming back to that diagram of starting with DNA, your permanent genetic code. DNA gets transcribed into pre-mRNA. Pre-mRNA contains both protein coding sequences and non-protein coding sequences, and those non-protein coding sequences need to be spliced out, so that's where splicing comes into play, and then you're left with your mature messenger RNA, or mRNA. And that mRNA, if it is correctly spliced, will produce the protein that your cell needs to survive or to be healthy or to do the function it needs to do. SKY-0515 intervenes at this step of splicing, and in the target genes of Huntington and PMS1, it introduces a small sequence that essentially triggers the cell, it signals the cell to destroy that RNA. So SKY-0515 has a dual mechanism of action. It lowers the amount of the harmful Huntington RNA, so I should say it lowers the level of total Huntington RNA, which includes both wild type and mutant Huntington. And it also lowers, to some extent, the amount of PMS1 RNA. And PMS1 is a protein that has come up as one of the key genetic modifiers that is implicated in the continued expansion of the CAG repeat that is inherited over time, which happens predominantly in neurons, which you heard a little bit about this morning also from Rachel. And I have another slide to go through that in a bit more detail. So this is all very familiar to you at this point, but Huntington's disease is caused by a mutation in the Huntington gene. This is a mutation that is inherited. It is generally inherited on one copy. We all have 2 copies of the Huntington gene, and is generally inherited on one copy. And this mutation is a repeat, which we call the CAG repeat or CAG repeat, which gets expanded into 36 or more CAG repeats, so someone with 36 or more is at risk of developing Huntington's disease, and someone with 40 or more generally will develop Huntington's disease. And the result of this expansion of the CAG repeat, because the CAG repeat is in the protein coding portion of the RNA, this means that translates into the protein. And this produces then a protein that is different, containing additional sequence that modifies the structure of the protein and essentially makes it toxic to the cell. And that then ends up eventually killing the cells. And in particular, this is one of the most vulnerable tissues. Our brain cells, or striatal, the striatum you may have heard about, is the region of the brain that is the most vulnerable in Huntington's disease. So the effect over time is a general shrinking of brain tissue. It starts in the striatum, but the brain overall, over long durations, gets smaller, and this affects movement, thinking, and emotions. So all of the symptoms that we think about when we think about Huntington's disease. So this is, again, coming back to the sequence of events of going from DNA all the way to protein, with a little bit more zooming in on exactly where SKY-0515 is intervening. So on the top there, you have a typical transcription to translation of protein. So you have your Huntington gene, mutant. In this case, this is the mutant gene. This is not really the right number of CAG repeats, but let's call it the expanded CAG repeat in exon 1. This is the pre-mRNA with the protein coding parts and the introns. And then you'll see this little pink box, which is something a little bit different, and I'll talk about that in a minute. And splicing then occurs, and it chops out all the pieces that the normal biology told it to chop out. And then you get your mRNA, and that gets translated into your protein. And in the case of the mutant Huntington, that gets translated into your mutant Huntington protein, which forms these toxic aggregates in the brain, which ultimately lead to cell death. When we add SKY-0515, that SKY-0515 interacts with the RNA at this position in the what we call intronic region of the pre-mRNA. But this sequence right here is actually a little bit like an exon. It's not normally included, but we strengthen that inclusion of that little sequence right here with SKY-0515. And when that pink box gets included in the mRNA, that sends a signal to the cell that this RNA should be degraded. And when RNA is degraded, it's no longer available to be translated into protein, and therefore you have a reduction of your mutant Huntington protein, and therefore a reduction of your toxic aggregates. And hopefully this helps to preserve the function of the brain and reduce the amount of neurodegeneration. And I wanted to share one slide of data from our recent phase 1 study, which was conducted in Australia in healthy volunteers. This was a 14-day study. It was studied at three doses. So we had 4 groups of subjects. So these are, again, our healthy volunteers. We have placebo, which is no drug. And then we have our low dose, which was 1 milligram, a mid dose of 3 milligrams, and a high dose of 9 milligrams. And what you can see here is the results of the RNA lowering in blood. So this is what we expect to see also in the brain, because we cannot take biopsies of the brain. But based on our early preclinical studies in animals, we expect to have similar lowering in blood and brain. And what we can see is we have a very nice dose response and a very nice range of lowering between 20 and 70% within this very narrow range of doses. So what this means is that moving forward, we'll be able to explore the safety and efficacy of lowering Huntington, and again, total Huntington, RNA, and protein. So I should also mention, we do also expect that this correlates to protein lowering, and we will be measuring protein lowering in both blood and CSF moving forward. But this time point was too early to look at protein lowering. So this is just the RNA. But we will be able to explore the efficacy and safety within a quite a nice range of lowering, between 20 and up to even 70% plus of lowering. So I have just, I think, this last slide on PMS1 that I want to talk about, and then just a little bit about our clinical status and next steps. So just coming back to PMS1, because it is, for us who have been working in this area, we've been hearing about it for quite some time, but it is a rather new concept to be targeting these gene modifiers that have come up as important for facilitating what's called the somatic expansion. And this is the continued expansion of your CAG repeat over the lifetime of the patient, which predominantly happens in the brain, in the neurons. So again, I just want to say that as we're all born, Huntington's patients are born with an extra number of CAG repeats in one gene, and the more CAG repeats that they're born with can correlate to earlier onset of disease. But in addition to that, what we now understand better is that in these vulnerable cells, such as neurons in the striatum, that CAG repeat goes from, let's say, 45 copies up to 100 or even more copies over time. And that's facilitated by these genetic modifier proteins, such as PMS1 or MSH3 or FAN1, which you heard about this morning. And so by lowering PMS1, we expect or hope that this can, to some extent, delay or slow down the expansion of the CAG repeat, which is really what is driving this very long repeat and very toxic protein in the later stages of the disease. So overall, mutant Huntington is a damaged protein caused by an expanded repeat, CAG repeat, that is continued to be expanded over time, and that continued expansion is facilitated by proteins such as PMS1. And those proteins are helping to expand the repeat, but by reducing that protein, we may be able to slow down that expansion. And so by both targeting PMS1 and the mutant and the Huntington protein, we believe we can have a synergistic effect in terms of modifying the disease outcome. Of course, this is all exploratory at this point. We are in the clinic, and we are doing the studies to determine if we can, in fact, do that. As I mentioned, we have completed our phase 1 in healthy volunteers. This was completed in Australia last year. We've continued that phase 1 in a small number of HD patients, and that is ongoing. Dosing in that started in January and is currently running for 3 months. And we are also happy to say that we've just received approval for our phase 2 study to initiate in Australia, and that is expected to start by end of Q2. This is, I should have mentioned that this is, you saw this morning also from Rachel, and I think you also saw it for those that attended Roche's talk from Peter. This is a standard clinical trial design where you start with your phase 1 studies focused on safety. In some cases, you can have look at early signs of efficacy, generally biomarkers such as the RNA lowering that I showed you. As we move into phase 2, we'll definitely be still very focused on safety, but we will also be incorporating various other endpoints, including biomarkers and clinical endpoints, as well as imaging, which you might have heard about today as well. And finally, as we then move forward into our phase 2-3, we will be essentially establishing the efficacy and confirming the efficacy of our drug. And that, I wanted to say, is in the work. We're starting to plan a phase 2-3 study, which would be a global study, and look forward to hopefully being able to talk more about that soon. So just as to summarize in a couple slides, SKY-0515 is a small molecule that is taken orally by mouth once per day. It spreads very well throughout the body, including into the brain, and it lowers the amount of total Huntington RNA and protein, as well as PMS1 RNA and protein. I also wanted to emphasize that SKY-0515 is reversible, so when you stop taking SKY-0515, it will wash out of the body. The RNA levels will return to what they were before, and therefore the effect can be reversed at that time in terms of the RNA lowering. I mentioned that we've completed the phase 1 study in healthy volunteers. We're very happy to have the ability at this stage to look at lowering levels of Huntington upwards of 70%. So far, no notable safety findings have been identified, but we are continuing to track this as we move forward into our further studies. We are dosing patients currently in Australia, and we are about to start soon our phase 2, also in Australia, and expanding into New Zealand, and eventually soon into rest of world. I wanted to, if you have not had a chance yet, please go by our booth and visit my wonderful colleagues, Maddie Mokhtarani and Tracy Hollowell. We have a lot of fun little giveaways, and we're happy to talk to you about any questions you may have. And lastly, I thank you all for your time. You can contact us with any questions at this email address. And just let people take photos. Go ahead. Okay. And then I think there was one more slide. Oh, no. Okay. We will, please, we have now live our phase 2 study on clinicaltrials.gov, so you can search for us on clinicaltrials.gov. Also happy to provide that code for you if you come by our booth. It was supposed to be in that last slide, but it didn't get added. So thank you very much, and happy to take any questions.
Hi. First of all, a comment. Thank you for this presentation. That was really simple to understand compared to other presentations, so you did a very great job. Thank you.
Thank you. I'm happy to hear that.
And I have a question about this. We already quickly discussed that, about the side effect of this drug that is also targeting PSM1.
PMS1, yeah.
PMS1. My question is, does it target also over protein, over gene? Because
other
you
genes.
communicate about this one, but probably there is off target or side effect, and how the medical agency are seeing that. Is it a concern?
How the regulatory authorities are seeing this?
Yeah,
I think that's a great question. So selectivity is a very important aspect of developing small molecules that modulate splicing. And we've seen that historically in other studies of small molecules. SKY-0515 was developed to be very selective for Huntington, less so for PMS1, so the modulation of PMS1 will be lower. And they are within the top-ranking targets of SKY-0515. So there is a number, and it's definitely dose-dependent, of course. As you go up in dose, you get more and more off-targets. And in this case, we're very happy to be able to have doses that lower Huntington at this levels without targeting too many off-targets and targets that would be of concern. Does that answer your question? Thank you.
With the fact that you're targeting PMS1, is there a reason that PMS1 couldn't at some point be a separate drug instead of having it combined with the Huntington lowering, because I know that there's concerns that lowering both copies of the Huntington protein might cause issues that we just haven't seen yet in these studies. So, like, is there a possibility that, like, PMS1 could be a separate drug that could be combined with a different drug later?
Yeah, that's a very, very good question. And absolutely, there is that possibility. And you're absolutely right. As I mentioned, the potency or the effect on PMS1 is not as strong as the effect on Huntington. And so if we don't want to lower Huntington as high, then we may want to think about a separate drug that targets PMS1. And the question around how much PMS1 lowering is required is also open. So these are all things that we are considering as we move forward with our development. Yes.
That's a really lovely presentation. It's Rachel Harding from HD Buzz. I had a question about exactly that. So we saw with branaplam that things looked very safe in the previous use of that drug, where in a non-HD context, Huntington lowering was tolerated. When we moved to people with HD, things went sideways very quickly. That trial failed quite spectacularly. Yeah. So, yeah, what are your concerns and considerations? Going 72% in healthy volunteers is very different from what we'd expect to see in HD, perhaps.
HD. Yeah, absolutely. I mean, we're very familiar with what has happened in that area, and we are of course monitoring this very closely. One of the key outcomes that correlated with the side effects and the toxicity in branaplam was the increase in NFL. So this is one thing that we're implementing very early. We have medical monitors, obviously, controlling on the patient side if there are any symptoms that could be related to some of the side effects that we're seeing with branaplam. We do have a different profile than branaplam, so that's very important to say. And while we don't want to, of course, consider us, out of the woods just because of that, we're being very cautious about how we move forward and making sure that we're keeping everything in mind. But we are different than branaplam.
What would be the best time to start taking SKY-0515?
In terms of stage?
Yes.
That's the golden question that I think we're all trying to answer. We talked a little bit earlier. So we are currently doing the studies in earlier stage. So for HDYO stages 2 and early stage 3. We are very interested in going earlier. We are talking to regulatory authorities, key leaders in the area about that question. I think it is definitely to be considered that the earlier we go, the more effective we can be. But we do want to make sure that we are doing our studies in a way that we can make sure that we can see the efficacy, but also maintain the issue around safety in the right way before we go too early in non-symptomatic patients. Yeah.
Hi. If the pill is taken daily, is there an effect that shortens like the trials? Because if it's taken daily, and the effects can be reversed as well, does that shorten like the period of time that the trials need?
So it's not about. So I'm trying to think of. So I don't think so. If I'm understanding your question correctly, the daily aspect of this is just how long the drug sticks around in the body and can do its function. But we're still essentially trying to maintain a steady state of the effect like other modalities are doing. They just might do it at different frequencies depending on the properties of those drugs. And it's the duration of that lowering or of Huntington or any other target or effect on a target that is ultimately going to lead to the efficacy of the improvements in the disease. So that's probably more a function of how much lowering do we need and for how long than on the frequency of the drug. Does that make sense? Is that helpful?
So I'm just curious, since it's kind of different with the Huntington's protein in people who have it and then people who are like considered healthy individuals and aren't going to develop HD. What is the significance of the results of the phase 1 trial of like the lowering of the RNA?
So that's a good question. So we all have Huntington RNA, and actually the mechanism of SKY-0515 is independent of it being mutant or non-mutant. So it does not work faster on mutant Huntington RNA than it does on wild-type Huntington RNA. So this is essentially a surrogate for us to understand what we would expect in a patient. It should be lowering the total RNA in a Huntington patient at the same levels. Does that address your question?
Yeah, I think so.
You can rephrase it if you have something else you want to ask.
No, that's all I have for now.
Okay. Yeah.
Thanks. You were talking about this compound's much more selective for Huntington and PMIS1. Mm-hmm. I was just wondering how you determined that. Was it looking at specific off targets in particular, or did you look at the whole transcriptome? Did you look at every RNA, and where did you look at those when you tested it? What model did you look in?
So yes, early on in development, we've done a lot of RNA-seq and testing, looking at the whole transcriptome, and we started in human lymphoblasts, and then we moved into also other species because we also wanted to understand when we're doing our preclinical tox studies how they relate to what's being modulated in humans, and I believe we did it also in iPSC-derived neurons as well.
Thanks.
Thank you very much, and we're at the booth for the rest of the meeting, so please come by and ask any more questions. Thank you.